Yeah, welcome everybody for our fourth annual in vitro software user meeting. Thanks for coming here, and also everybody that joined us online for today's session. We already had a great workshop around VivoQuant scripting yesterday, which many of you attended. That I heard was very successful and was well received. And today we wanted to present to you a little bit of our work that we have done over the last year, as well as give you an outlook on what we as the software group at Invicro are focusing on, and give a couple of our users also a chance to present on their work, which very likely have similar problems that you have and used Invicro solutions to solve them. And I also hope that we have a lively discussion about things. So please ask questions. This is also more like a workshop where we can also go into more details of things that are of interest to you. So please feel free to come up with questions. We are also, that's not working yet. Yeah, we are also celebrating actually today the 10-year anniversary of Invicro. To the day, 10 years ago was our incorporation of the Invicro LLC, and we are having some internal celebrations also. But we also wanted to share this with you, and we should have a drink on Invicro later at the bar downstairs. I hope you brought the champagne or something. Champagne if you want. So, and with this, I would like to pass on to Bill for the introductory talk. Thank you.
Thanks, Christian. And thanks everyone for joining today, both in person and online. We are excited for a robust discussion around, as Christian said, how users both internally at Invicro and externally at pharma and academic institutions are collaborating with each other to solve these challenging data informatics problems in an effort to answer these even more challenging biologic questions and advancement of their drug development programs. As a play on our 10-year anniversary, I thought it'd be interesting just to look at how we got here. Our marketing group did a nice job kind of recapturing a brief timeline history of Invicro, and we've gone through a lot of change, or especially over the last couple of years. From acquiring companies, merging with groups, specifically around the scientific and operational capabilities of all these teams. And, most recently with the acquisition by Invicro by KM, really wanted to just highlight on a couple comments on what that means moving forward for our users and all the scientists that we work with day to day. Konica, after acquiring both Ambry Genetics and Invicro, really set out to form a advanced precision medicine group that is combining world expertise across different bioinformatics, quantitative biomarker disciplines, from genomics, histopathology to radiology. And Invicro, as we look at how we support and fit into that platform, we really view ourselves as a key data analytics technology driver of how to best leverage the current practices in managing data, viewing and analyzing data, reporting on that data from a variety of different modality types, whether it be a variety of modalities within radiology. With Konica Minolta, we've been advancing into the histopathology space more, and how best we can collaborate with our Ambry colleagues, as well as, I think, the quantitative biomarkers at pharma that are producing other data sets that groups are wishing to merge and to analyze in a cross-modality setting to answer these complex questions. And so the tools and the people that we have organized across the enterprise will continue to form into a group that will be made available to you as customers from a deployed software standpoint, as well as the continued engagement we have with many of our customers in the pharma data analysis space for the contract research services we offer as well. So very exciting times here at Invicro, and we continue to make investments in our company to stay at the cutting edge of how we think about data handling, data processing, and general data analytics practices. A little more specifics on in vitro, we continue to stay as an organization laser focused on radiology and increasingly some histopathology-based assays and the data handling tasks, data processing tasks that come with that. What may have gotten hidden from the large acquisition from KM was us joining forces with the Imanova team and their scientific group and operational capabilities, their site capabilities, Core Clinical, who their team has a wide breadth of experience in late stage imaging core lab services, particularly on oncology, but other therapeutic areas. And so the challenges that our software team and the image analysis group here at Invicro kind of jointly face to help support the wide array of different types of data, again, from a data handling, data processing, data analytics standpoint, are only getting more complex. And we're looking forward to exploring how best to provide solutions, not just internally, but working with you as well, as we certainly appreciate and enjoy all the scientific collaboration with you as customers as we try to jointly build out these solutions to meet each other's needs. And how we envision the future building and supporting the wide array of these types of data sets is really continuing focus on the core software platforms that we develop in-house. But I think you'll hear a great theme today of how our software technologies can be incorporated into a wider picture, a larger footprint or ecosystem of different software tools for handling these different types of data sets that may fall within the radiology, histopathology category or complement to such. So you may be familiar with this diagram of how, IPACs currently in VivoQuant as a platform handle a variety of different preclinical and clinical data workflows, whether it's from a data sharing standpoint, data transfer standpoint, or from the nuts and bolts of viewing and analyzing the data. We continue to see IPACs as a core platform to manage and be a content management system for all things data related in imaging trial. And how we see the roadmap building out is where we can make investments on improving integrations to not just our own software, but other tools that groups are using so that IPACs continues to be a center hub of the wheel, so to speak, of how you think about managing your image data. With that being said, the current environment is very interesting in how the landscape is evolving of how groups are thinking about managing not just image data, but all types of data within pharma. And as you can maybe see from a few slides ago, even at Invicro and working with our Ambry colleagues and our KM colleagues of how can we best think of IPACs as a component of a larger data management strategy so that groups, when you have this single question of, can I look at a correlation of data output from a variety of quantitative biomarkers, that the technology and the infrastructure is not limiting to how you're accessing that data, how you're interacting with that data, how you're analyzing that data. And so that is really a core focus, and I think will be a theme of the year in how we work with groups, our current customers, our hopeful new customers we don't know yet, but hope to meet soon and even solve challenges internally. And I think what continues to be such an exciting time at Invicro is the symbiotic nature between the problems that are presented to us through our contract research business and the problems that we interact with our customers using our deployed versions of software. We really see that line getting more gray in how we think about supporting these data handling, data processing, data analytic challenges. And so with that, I think, it's a great way to set the tone for many of the themes you'll hear in these talks today. I'll try to shut up quickly because we have a great lineup of speakers that will be touching on a variety of these topics. But, I think it's important, especially at a day like today, to really make sure that from Invicro, you realize how much we thank our relationships and our collaborative time with you as our users, and also our own team. So there's a lot of people here today, hopefully, that users, customers get a chance to meet. You see myself, TJ, Rhonda, Melissa as really been the main interface to you in a lot of these projects, but there's a huge team behind us that helps support a lot of the product development, the features development, general application support, technical support. And so it really, I hope everyone gets an opportunity to interact with the entire team here. That's from our side. And then again, we really should be thanking our users. You help drive our success. So with that, a little play on one of the first marketing campaigns we ever had at the company. If anyone caught it, I think it was back in 2013, but it was highlighted by a phrase, "See more." So let's help each other see more today, one voxel at a time.
So, hello everyone. Hope everyone had a good lunch. So my name is Yannick Cadare. I'm a senior software engineer for Invicro. I work on the VivoQuant team. So I'm here today to introduce the next speaker, who it's been a good collaboration that we had again this year. I think we came up with a good teamwork with their team and our team to really come up with what I think is a great workflow that really leverage a lot of the tools that we already had at Invicro and a lot of the software that we had. And this collaboration also allowed us to bring up the next level of analysis of ultrasound that we had in VivoQuant. And it's been really a neat addition to the software, and it's really a step forward to us to move to more ultrasound analysis. So without further ado, Raymond Chang from Regeneron Pharmaceuticals.
Let me see. Do we have a point? Do I just use my hand? Oh, there you go. All right. See. Okay. Hi everyone. Thank you for inviting me today to give a talk about what we do with what we did with the ultrasound. First of all, when we first started with the projects, one of the things that I initially thought about was the difficulty of quantifying ultrasound and the amount of noise that usually present in ultrasound. So I was kind of doubtful at some point whether or not it's doable. Well, obviously, I was shown incorrectly. It could be done. Okay, so let me introduce you from our CV group. I'm coming from a Regeneron, they call it therapeutic focus area. We focus a lot on a lot of cardiovascular modeling, a lot of toxicology modeling. And we have a whole shoot of stuff that we usually do. And one of the things that we use a lot is really ultrasound. So in order to kind of give a high throughput of all this data, we have to kind of figure out to streamline the process. So as you can see, we have a lot of things going on. And ultrasound play a big part of what we do, which of course, cardiovascular ultrasound is probably the easiest thing to do. However, the huge problem that if you can see is that in the ultrasound system is the analysis portion, which I will show briefly. So what we use most, and it's really one of the essentially top of the line in vivo imaging for small animals for ultrasound, is the Vivo 2100 system. And you can upgrade to 3100, but in our case, 2100 works what we want because, a button works better than a touchscreen, and we do a lot of mice. So we try to do it as fast as we could, and usually we can do pretty quickly. It's just a matter of getting the right sort of data. And if you can see in the in your bottom left here is the Vivo 2100 with the imaging bed, and then here this is what you're looking at. Is really the M mode is our focus. M mode images of a left ventricle of a mouse. So what is the main problem with ultrasound when we come to this? Now, last year we're presenting the B mode data. B-mode analysis. And this year is essentially the second part. We're presenting data related to what we call M mode or motion mode. And I will briefly explain what motion mode means. If you guys are not familiar with ultrasound, and I will briefly show what M mode usually show and how it is related to an image of a heart. Now, one of the problem with ultrasound is really data bottleneck. And we do a lot of animals. As a result, you have backlog of data, and sometimes it takes months to analyze. So imagine doing 120 animals, 7 time points, and you're going to sit there and doing it for a long time, which I did. It took about a whole 2 months to do all of that, and it is not fun. The other one that we basically, the whole process started is the data integrity. So we want the data to be not only acquire, but we want to be able to keep and update the data. So this is why IPAC comes in. So we initially acquire IPACs, and then we kind of get into VQ, Vivo Quant, and from Vivo Quant, and that's where all the scripting of the M mode and B mode come into play. So if you look at the right side, that you could see what M mode would actually look like. I will explain what this jagged line is if you're not familiar. And this is the diastolic and systolic frame of a same heart, with the exception that it is on a lengthwise image. And this is where we do get most of the data. I mean, the data analysis, and it's fairly simple. It's just that the image qualities and how we can go about grab these data is really one of the major challenge. So give you the perspective. So I basically go out my way and tally out how much time we spend on analyzing these data. So if you look at each mice and different model, this is the mostly genotyping pharmacology challenge. This is the infarct or heart attack model, and then congestive heart failure model. You can see that the amount of time per mouse, total time, is about average 30 to 45 minutes, 30 to 40 minutes per mouse. So take that in perspective. And if you look at what I pull out, and I actually did the math yesterday, that we did about 48 studies, about 2,700 mice we scanned. So we spent about 63 days just to analyze all these data. And we spent approximately scan about 55 times of a mouse. We actually scan 55 mice per studies. So that's pretty intense, because if that's what you do, and that's going to be a very, very hard work to do. So if you look at the M mode, how the M mode related to a LV or a left ventricle. And if you look at the heart, and we actually usually go from the base to the apex, and we usually acquire the data at the middle portion of the left ventricle. So if you look at this is one of the MR representation of a left ventricle, and you see those 2 little dots. That is what this is a pulmonary muscle. It's an attachment of a muscle that is from the base to all the way to the apex, where it controls contraction. And we use that as a landmark. So when we image a heart, we want to see that. And if you look at the side, if you see this jagged line, really the top wall is the anterior wall, the bottom wall is the posterior wall. That little jagged line there, that is an apex. That is a pulmonary muscle. That is critical, because if we could see these, we usually hit about the same part of the heart. And if you don't, so this is one of the critical points. Ultrasound is really landmark-based imaging and very focused, specific area. So for us, we kind of leveraging IPACs in that respect. And how we usually do it is that in a broader sense, we do most of the study, and I stole it from Vicro, then I kind of modify it. So we do a study, and then we acquire the images, and we have an option to either allow in Vicro to pull the data for other studies, or we have an option of doing further clinical trials if we want to. Haven't happened yet, but there is a potential. And then the data, and then analyze. And in this case, we run it through the script. And from the script, we compile the data, and we're going to push it back to IPACS, and then we leverage an IPACS to pull all the quantification that we necessary for our analysis for our trials. In a deeper dive, if you look at it, this is how we set up. The whole thing was done. You don't really have to do much of a data transfer. The only thing that we prefer to do is that the data, every time we get a data from a Vivo system, you have an option to pull data directly to link it to IPAC. In our case, we decided they want to do it through our own server, and all the data transfer, auto transfer, will be done from the server. So the beauty of this is that every time we do the data, we get a data and we just walk away, and the data will be in the server, will be in the IPACS, and then we could do everything through IPACS. And a data transfer, and then go into the IPAC folder. And then once we go in there, we run everything through VivoQuant. We run the script, and the script allows us to do the semi-automatic segmentation in a batch format. So once you run it, we run through all the animals. From that, we then can allow to see a QC. We can see how well the segmentation is. From then we could then modify, and then we push it back to IPACS, and then we then leveraging IPACS and pull all the data for further analysis. So you can essentially do a study, like you set a study up, you walk away, and you have multiple time points. The data will automatically generate into IPACS, and then you don't have to worry about losing data. So then again, I will show you that I just briefly explain what the MO data is. And one of the things about this data is that if you look closer, it is a scroll-like image. So it's a long image. So you cannot imagine yourself analyze all of it. So we usually do a couple of cycles. So in our case, we want to select a portion of a cycle. So the script allows us to do this, where we select specific portion. The other thing that we want to be able to do is that we try to avoid the respiratory cycle. So you see here, the distortion coming from the animals like breathing in and out. So if you quantify that portion, your data will be less accurate. So we want to avoid that. So allow us to select the image. And once the data is selected, the script that, oh, it actually moved. So once we select it, the script allows us to do putting line into the images. So the line, to generate the line, one of the things that Yannick and I believe Mohammed came up with it is that by putting C points, and the C points allow you to select, triangulate a specific local maximum point. It will propagate the line. All you have to do is, if you imagine this, you click top line and top and bottom of the line, it will automatically propagate that line, create single side of the wall, and you do the next two, and then what happens is that it will give you a anterior wall and posterior wall, and then these lines are automatically generated based on the attachment to the line wall, essentially, and give you the all the necessary data for doing analyzing a cardiac function. So surprisingly, I found that these lines are very critical, and I will explain why briefly, why it's so critical for these, specifically for MO data. And once the data are analyzed, they are then stored in IPAC folder. The format that we have is that usually we have a raw data, and the raw data, and then after we do an analysis, go into a process data or _P data. In there, we can store both the B mode and MO data, and you could pull them separately depending on your need. And I assume that if anybody can take this, use this system, probably very similar design will be used. The other thing that I want to point out is that the heart rate and cardiac output, the reason is these two values are heart rate driven, and also is depending on your unit or your ultrasound system. So during your acquisition, you have to set your system in a certain way. Otherwise, the script will not be able to pull it out. And so it's all depends on how you set your system. So if you don't see it because you didn't set it in your ultrasound unit, so just to be aware of that. And I found them like, why don't I get these data? Well, because you didn't set it, because the metadata doesn't have those information, so we can pull it out. So it all depends on me and us. So the easiest way for us to test the system, we use the same data set like last year, is that we're doing what we call a stress test. And to do stress tests in little mice, you don't just make them like, doing the running. And we give them an injection of isoproternol. Isoproternol essentially stress your heart, make it move faster. But the other thing it didn't do is that it will not change the size of your heart. So in other words, your cardiac mass won't change. And that's critical because we shouldn't see the changes. And now, do you actually see it? Well, I'll show you quickly. So here we give a ultrasound, initially baseline. We give a dose of isoproternol, and then we scan them 15 minutes later. And then you could make the whopping change, a lot of motion in the heart. So here, this is one set of data I used, and actually one of the analysts, she's actually sitting right there. So here, if you compare the left side is the algorithm base, basically the script base. The other ones is we actually go out and trace it. But the beauty of this is that not only that it mimic both sides of it, it actually detect the same outlier in both, which means that the system works. Not only it works, it actually detect the outlier. Like if it didn't detect it, then I'm kind of concerned. And then similarly, and this is the ejection fraction, and you see the same outlier in both cases. I think it's wonderful. And then you see something, you see the wall a little bit variable, and I'll explain. The variability is interesting because if you do it manually, you have to do it, actually put one line at a time. In the algorithm, you don't even have to worry about that. And that's the beauty of it, and that's really a huge benefit. Now, when I talk about the mass, right? Lo and behold, they both have similar result. Not only that, there's no difference. In other words, we're using the right kind of test because the mass, if the mass change, then we have a problem because the estimate of that mass shouldn't change because the effects are temporary. So it does what it's supposed to do. Awesome. And here, if this is generated through VQ, that's VQ, the algorithm base, and this is traced manually. You can see that they're pretty similar. This is the animal that actually went through isoproternol. So their heart like really pushing really hard. And here, this is, all these are done with the same animal. So animal inject baseline, and they inject and scan it again. And you see that the changes, and the top is a manually segmented. You see the wall get thickening, but it didn't change the mass. And that's important, that's the good test. Similarly with the baseline on the algorithm, and you see that they're very similar result. And this is really the important test. And then what we did, then we do a regression on these. We're comparing the two methods, automated or algorithm based, and then the manually segmented. So the top one, okay, here, the top one and the bottom one, the top is systolic. That's when the heart really when we contract, and diastolic when the heart expand. This is a volume estimate. It looks almost like 1-to-1, and which a lot of our computation is based on the volumetric assumption. So it does what it's supposed to do, and the same thing with the ejection fraction. It works out pretty well, even fractional shortening. Which if briefly, the B mode fractional shortening is tougher to do. If you were here last year, and we actually explained why it wasn't as good, and we kind of improve it over time. However, if you look at the mass, that's where the issues is. And the mass has to do with, again, it's a line placement. And I think at the end, I'll show you a graph that clearly show the line placement is one that played a role. And then the wall, kind of iffy, but it's, again, there's a line placement issues that it doesn't exist in the algorithm, but it does exist in the manual segmentation. And I have been struggling to figure out how to really display this result. So this is the R-square, the goodness of fit analysis. You can see those two is clearly an issue, right? If you compare the two methods, a manual algorithm based and manually based, you can see a huge variation, the variation between two methods. And the least accurate is really on the wall and the mass. So the question is, which I'll explain why, the question is why. So before I do that, I want to just give you a brief, review what we have done. So we found that it works quite well, and it works really well with the conjunction with the B mode. Similarly, the problem that we have again is how well we could fix the problem of ultrasound in terms of image qualities, because really, right? Sometimes they garbage in, garbage out. If you do a terrible job on the imaging, well, you're gonna probably get a pretty terrible data. So because ultrasound is so noisy, we gotta figure out how to really kind of fix the image. And how do we do it? Well, we could either figure out best way to acquire the data, but based on what Yannick and the team, they said that we did the best images. So I don't know how much we can improve. Maybe they have a better system to do the stuff, to do the work. The other one is how do we possibly correct the noise? I mean, the problem with ultrasound is that the blood going through the heart, the blood creating these ultrasonic shadows. So they create these spackle noise. A spackle noise, when it flow into the muscle, it creates these blurring line, and it's really hard to really get around it. Now, it could be a good model for doing the AI segmentation. I think that is like a lot of potential. I hope one day I don't have to sit there and do it all day. So if I just put it in the AI, let the AI does all the work, that'll be awesome. So now the other improvement that we see that we kind of make Rhonda and Yannick do it is to put a B mode Cena loop as a reference image. The reason is, if you look at the M mode image, the M mode image is pretty benign because you look at it, it's like this wavy line, and sometimes on the top line you have multiple lines. You don't even know which way is the wall. So having a reference image is very important. So this reference image now, our newest script is allow you to see where we actually drop the line and where the top line wall is. It's so critical because if you just do this, you don't even know which is the line. So which is why it's so important, because your C points are based on where you put the line, where you put it bottom and at the top and the bottom of the line. So I think this is add a lot of values. And I think that we're going to do the same thing on the B mode as well. Now, before I go to a really nice slide that Bill wanted me to show, I'll show you. It's a good one. Not this one. This is awesome one. This is the one I'm going to show is good, but this is awesome. So I want to, of course, I'm going to acknowledge y'all. We have done a lot of work. I mean, last 2 or 3 years, we did a lot of work with everybody. So of course, our director with our blessing, and Michael, that he gave the B mode talk last year. And of course, this is my colleagues, which by the way, he actually was in Jeopardy. He actually in Jeopardy. I'm not sure who actually seen the Jeopardy, but he was, he won some money, but he was cool though. He was in Jeopardy. That's real. That's not Photoshop. And okay. And this is Visual Sonic team. Andrew Nettles, and then Stephen Butter, and then Terry Conti will help us do all the really bridging and allow us to get the source code from InVicro to work on this project. Otherwise, this is not even possible. It would be very hard. And of course, the InVicro team, and I want to point out Bill, awesome pictures. The day he was playing baseball, and then Rhonda, of course, Yannick. It took me forever to find the pictures. The Bill was easy, but it's hard to find Yannick. I was surprised I could find him on his Facebook, but awesome. And then our Regeneron team, and we have been bugging him to do us a new version of VQ. He was really patient because I have like 5 different versions of VQ in my computer, and he was patiently download every single one. And I think that before I do that, I'm going to show you this. Why it works better. It's this slide. My initial idea, I want to put this in the front, but my boss told me that, hold off. All right. I talked to Bill and told Rhonda, I said, this is a good slide, and I know it's a good slide. The reason it's good is that it clearly showed the system work. Look at this. I compared the manual versus manual. So 2 different people analyzing using manual analysis, and you do a regression, lo and behold, the mass and then the wall looks terrible. Now, if you look at the algorithm, it looks not terrible, it's pretty good. Now, the reason is the line placement, because if you think about it, an individual put the line differently. The algorithm, the line placement is completely, you don't even have to do anything because it all determined by the peak and valley of your end mode, of the line generation. So I think that's a big plus. And I think if we could figure out to really further enhance, reduce the noise, I think it would be really, really a good system to use if, solely if that's what you do for ultrasound. Thank you. And I'm open for questions.
I always feel really sorry for ultrasound people having to present freeze frame images because it always looks great on the screen moving. And then you snap something for a slide and it looks all noisy, right?
They're noisy.
But I'm thinking with the VQ and the IPACs, perhaps is there an opportunity to sort of analyze multiple cycles and average?
You could, yeah, you could do that.
So it might
And
be
that's,
practical.
yeah, the whole idea is to, could we find a way to averaging these frames and then shrink it, and then you kind of find a way to reduce the noise. Unfortunately, I'm a little bit more different because in the B modes, if you look at the B modes, really the noise is solely coming from the blood. So you can essentially fix a lot of it by, a lot of image averaging. The problem with M mode is that sometimes the ultrasonic distortion causes the line to break. Because when we do this, right, we have the seed point allow you to find local maxima. If that's the line breaks, that line won't be straight. The line will be like jagged. So you have to kind of clean it up. And I think that's a serious problem. It's really no easy solution. But I hope someone could come up with something that would really make the whole, the process and also the data quality better. No question? All right. Thank you.